Now come to the next. Agrobacterium, right? Agrobacterium tumefaciens. Agrobacterium tumefaciens, by transferring the tDNA, that is transfer DNA, into the plant cell, it results in what will happen in that case? The piece of DNA which is called the tDNA to transform the normal plant cell into a tumor cell, right? And directly these tumor cells to produce the chemicals which is required by the pathogen. So this bacterium invades the plants at the site of wound, right? And what will happen? Transferring them and nearby the cell to form a tumor which is called the crown gall, right? Which is called the crown gall. Clear? So when the bacterium contacts a damaged plant cell, it delivers the transfer DNA or the tDNA fragment from the plasmid into the host cells, right? What will happen? That integrates at a random position in a plant cell chromosomes, right? And the tumor inducing plasmid has now been modifying into the cloning vector, right? It has been now modifying into the cloning vector. So we can say that that plant cell results in the tumor formation, right? Which is no more the pathogenic effect. Now the plant but it is still able to use the mechanism to deliver the genes of our interest, right? So that is the vector for the cloning genes in the plants, right? That is Ti plasmid of the Agrobacterium tumefaciens. Now come to the next, that is the virus used to deliver the desirable gene into the animal cell is called the retrovirus, the reovirus, both or none, they are known as the retroviruses, right? They are called the retrovirus. Come to the next. DNA is hydrophobic, hydrophilic, lipophilic, none. Phobic word is used for the fear factor, philic that is loving, right? And DNA is the water loving, right? DNA is the water loving, that means the DNA is the hydrophilic, right? DNA is the hydrophilic. So the answer is Bombay. Come to the next. Efficiency of the bacteria to take up the rDNA, that is recombinant DNA, increases by the concentrating solution by what? The monovalent action, the monovalent anion, the monovalent cation, divalent anion or divalent cation, right? So the answer is the efficiency of bacteria to take up the rDNA increases by the concentrating solution by the divalent cations, right? You have to learn it actually. To insert recombinant DNA directly into the nucleus of the host cell is called the incubation, no. Microinjection, yes. Transformation, no, right? Because we say that insert rDNA directly into the nucleus and it is possible just through the microinjection. Even in case of the art, right, that ICSI, in the process of ICSI, right, we also use that microinjection to enter the sperm directly inside the ooplasm, right? So that's why that incubation, that is the time duration between the that is the time duration of appearing of the first symptom after entering the microorganism in the body, right? That is called the incubation period. So it's not like that, right? So the answer is the micro injection. Come to the next question. That gene gun to introduce gene into host cells is most suitable for, we already studied in studied about that gene gun in previous question, right? That it is most suitable for the plants, right? It is most suitable for the plants, right? So the answer is A. Even in NCERT, a line is mentioned for the gene gun that other than the microinjection method, right, which is suitable for the plants, which is suitable for the plants, right? Cells are bombarded with high velocity micro particles of gold or the tungsten coated with DNA in a method, right? They are known as the biolistics or the gene gun, right? So these are called the gene gun, which is most suitable for the plants, right? Actually in that question that most word is used, that's why the answer is the plant, right? Now come to the next question. 23rd question. In fungus, isolation of DNA is done using, right? What is that? That is the method for the isolation of the genetic material, that is DNA, right? As we know that this can be achieved by the treating the bacterial cell or the plant cell or the animal cell with some enzymes like the lysozyme. That lysozyme is used for the bacteria, right? That cellulase used for the plant cell, that chitinase used for the fungus, right? So the answer is C, because in question it clearly asked about the fungus, right? Come to the next. DNA is precipitated by using, actually that process is known as the spooling, right? That process is known as the spooling, and in that process we use the chilled ethanol, right? We use the chilled ethanol, so the answer is A, right? So the answer is A. Now come to the next question. The gene gun is also called as the microinjection, the biolistic, both A and B, none. We already studied about that. The gene gun is known as the biolistic, right? Come to the next question. That is enzyme used to remove the proteins from DNA. That is the protease is used to remove the proteins from DNA, right? So the answer is Bombay. Cutting and joining of the DNA are performed respectively by, we already studied that for the cutting we use the restriction enzyme. That's why restriction enzymes are also known as the chemical knife, right? Or the molecular scissors, right? Or the chemical scissors, right? And for the joining of the DNA, we use the ligase, right? We use the ligase enzyme. So these kind of enzymes are known as the DNA ligase enzyme, which join the DNA, right? So the answer is restriction enzyme lyase, no lyase restriction enzyme, no restriction enzyme, and the ligase, right? Exactly, right? So the answer is C. Ligate, that means the join, clear? Polymerase chain reaction is used to, that is PCR, right? That PCR is used to insert the rDNA to amplify the DNA to obtain the product of recombinant DNA, both B and C. So to amplify the DNA sample, right? To amplify the DNA sample, we use the polymerase chain reaction, right? Actually the question is now, what is that PCR, or we can say, actually this is very important process, right? And number of question asked through them, so we have to learn it properly, clear? Polymerase chain reaction, why we perform that polymerase chain reaction? To amplify of the gene of interest by using the PCR. That means we want multiple copies of the desired gene, right? In this reaction, the multiple copies of the gene or DNA of interest is synthesized in vitro, right? In vitro by using two sets of the primers, right? By using the two sets of the primers. What are these primers? These are the small chemically synthesized oligonucleotides actually, right? That are complementary to the region of the DNA. That in simple language we can say it increases the length of the DNA, right? It increases the length of the DNA and the enzyme DNA polymerase. Clear? Now what will happen here? See, in that process, these three processes are used here, right? The denaturation, first we separate the by the heat we separate the DNA strands, right? We start with a sample to we will start the sample of DNA to be amplified, right? And add 4 nucleotides and the enzyme DNA polymerase to it, right? See here what will happen in that case. We'll start with a sample of DNA to be amplified and add 4 nucleotides and the enzyme which is used, right, that is DNA polymerase into it. DNA polymerase to it, right? Then what will happen? Normally in vivo, right, the DNA double helix would be separated by the enzyme helicase, but when we talk about the PCR, that is in vitro, that means outside, right, these strands are separated. These strands are separated by heating, right? These strands are separated by the heating to 95 degrees centigrade for 2 minutes, right? Just for 2 minutes, which breaks the hydrogen bond, right? Which breaks the hydrogen bonds. Clear? Now what will happen? DNA polymerization also requires the short length of DNA, right? As we can see that is by the heating at the 95 degrees centigrade we separate that DNA, right? So what will happen now? DNA polymerization always requires the short length of DNA, right? That is it's just about 20 base pairs, right? It is just about max length of 20 base pairs, that is 20 BP long, right? And these are called the primers. These are called the primers. Clear? You can see here when we add the primers here, what will happen now? In vivo, that means inside the body when it's natural, right? The primers are made during the replication, right, by the DNA polymerase, right? But when we talk about the in vitro, right, but in vitro they must be synthesized separately, right, and added to this stage. Clear? So that's why after the preparing of the primers, we can add these primers here, right? And the DNA must be cooled to 40 degrees centigrade, right, to allow the primers to anneal, right, to anneal to their complementary sequence on these separated DNA molecules, right. So here we need the at the 40 degrees centigrade temperature we need to cool them, right. After heating we need to cool them. Then what will happen, that DNA polymerase, right, that DNA polymerase can now extend the primers, right. See that DNA polymerase, and DNA polymerase is different type of DNA polymerase here, but here the specific DNA polymerase is used, that is Thermus aquaticus, right. That's why it is also known as the Taq polymerase. That DNA polymerase is also known as the Taq polymerase, right, that is Thermus aquaticus, right. So it now extends the primers and complete the replication of the rest of the DNA. So you can see here, right, when we separate these strands, primers allow, right, separate these strands at the 95 degree centigrade, cool them at the 40 degree centigrade, right. Primers allow, and with the help of the Taq polymerase, what will happen, the chains are here, the number of the length of the DNA will be increased, right. So the enzyme which is used in the PCR, it is believed from the thermophilic bacterium which is called the Thermus aquaticus, right. The name of the enzyme is the Thermus aquaticus. Clear? Thermus aquaticus, which grows naturally in hot spring. Clear? It grows naturally in hot spring at a temperature of 90 degrees centigrade. So it is denatured. So it is not denatured by the high temperature. Clear? So simply in optimum temperature is about 72 degrees centigrade. The mixture is get heated to this temperature for few times, right, for few minutes, to allow the replication to take place as quickly as the possible, right. So what will happen, the each original DNA molecule has now been replicated to form two molecules, and in that way the number of DNA molecules get double. That's why we called it the chain reaction, right. And in PCR we need that PCR is run for 20 to 30 cycles. Clear? So that's why this is all about the PCR. Remember, the name of the DNA polymerase enzyme is Thermus aquaticus, which is used in the PCR, right. And remember about the temperature, at what temperature the denatured, at what temperature the annealing process is there, right. Now come to the next question. Thermostable DNA polymerase is obtained from. That thermostable DNA we already studied in the PCR chain reaction. Its name is the Thermus aquaticus, right. Its name is the Thermus aquaticus. Clear? So which is also known as the Taq polymerase in the PCR reaction, right. Come to the next. Below are given steps of the amplification arranged then in the proper order. See here, we already studied that first is the denaturation, second is the annealing, and third one is the extension, right. First one is the denaturation, so DAE, the sequence is the DAEA, that is amplified after that, right. So DAEA D, that is denaturation, annealing and extension. Denaturation, action, no. So the answer is the Bombay. Clear? That is denaturation, annealing and the extension. Now come to the next. Stirrer in bioreactors performs which function? It helps in the mixing of the contents, it increasing the oxygen availability, both, or maintains the salt and vitamins in the reactors. So when we talk about the stirrer, right, that is like that, right, in the bioreactor. So it helps in the mixing of the content as well as increase the oxygen availability, right. So the answer is the C, right. Come to the next question. Bioreactors with curved base but not having the bubble mechanism, right, it is usually present in. Now the question is, what is that bioreactor? This bioreactor is it acts as a vessel which provide the optimal condition for achieving the desired product, right. It is used for the for achieving the desired products by providing the optimum growth condition like the temperature, the pH, the substrate, the salts, the vitamins, all these things, right. So these are called the bioreactors. There are two types of the bioreactors stirred which is used. First one is the simple stirred tank bioreactor, right, and second one is the sparged stirred tank bioreactors through which the sterile air bubble are sparged. You can see here these are the air bubbles, right, which are used in the sparged stirred tank reactors, right. So in the simple stirred tank bioreactor, not having the bubble mechanism, right. So the answer is A. Clear? EFB stands for the European Federation of Biotechnology, Eco-Friendly Biotechnology, Environment-Friendly Biotechnology, or the so according to that, this is actually this is a organization, right. Its full name is the European Federation of the Biotechnology, which define the biotechnology as the integration. It defines the biotechnology as the integration of natural science and organisms, natural science, organisms, cells and parts of their and molecular analogues for the products and services, right, for the products and the services. So this is the actual definition of the biotechnology according to EFB, that is European Federation of the Biotechnology, which is also given in NCERT, right. So the answer is A. Now come to the next question. Autonomously replicating circular extra chromosomal DNA is known as the cosmid? No. Phasmid? No. Plastid? No. It is called the plasmids, right. It is called the plasmids. The extra chromosomal DNA is known as the plasmids. Our DNA was prepared using plasmid of, right, that is first, it's not I, that is Roman number, right. That is the first recombinant DNA was prepared using the plasmid of the Salmonella typhimurium, right. Salmonella typhimurium. That first rDNA was prepared by the using plasmid of the Salmonella typhimurium. So the answer is C, right? Come to the next. The restriction enzymes were used by the Herbert Boyer and Stanley Cohen, right? This is the direct question and you have to learn it, right? So come to the next, that first rDNA was prepared. The first recombinant DNA was prepared in the year of 1972, right? It is the direct question, so you have to learn it. Come to the next. Plasmid DNA acts as vector, vehicle for the rDNA, both or none. So plasmid DNA acts as a vector and acts as a vehicle for recombinant DNA also. So the answer is the both, right? So the answer is the both. So answer is C, right? Come to the next question. The restriction endonuclease Hind2 acts on the specific sequence of the 6 base pairs, right? This is also the direct question. That's why we say that the GARD, TTC and all these things, right? So these are these acts on the specific sequence of the 6 base pairs, right? Come to the next. What are in E. coli, that we also know that E stands for the genus, CO that is coli, that is species, right? That R denotes the rough strain, that is staining, and that one represents the Roman number in order to form, right? That one represents the Roman number in order to form. What does R in E. coli represents? So when we talk about that, we know that E represents the genus, that CO represents the species, that R represents the staining, that is rough staining, that one which is the Roman number, in order to form. Clear? So now that R is the gene, the species of the prokaryotic cell, no, species is the coli, name of the strain of prokaryotic cell, exactly, right? So the answer is C. Come to the next. That R in eco R. Come to the next question, that is gel for the electrophoresis is composed of, we know that gel is the agar-agar gel, right? So it is composed of the agar-agar. So inulin is cancelled, plasmalogens cancel, then agar-agar is the answer, right? Come to the next. That DNA is dash charged, hence move. We know that the DNA is the negatively charged particle, so we cancelled both of these, right? And hence moves towards the anode, right? They move towards the anode, not the cathodes. That's why the answer is Bombay, right? Come to the next question. The DNA fragment with dash size moves slowest in gel electrophoresis. As the size is largest, then it moves slowest, right? So simply the answer is the Bombay. Clear? So in that way, the principle and the processes of the biotechnology is completed in the form of the questions, right? Thank you.